Welcome to my channel Adhyan with Flow. In our previous video, we already completed non-cyclic photophosphorylation. Yes guys, we already done non-cyclic photophosphorylation. Look at here. This is the light reaction. This is the also we call it as a Z scheme. We also call it as a non-cyclic photophosphorylation. So this is the whole mechanism of non-cyclic photophosphorylation. How we can say this is a non-cyclic? Because electron pass here to here, then here, again this, and again here. This electron transport one photosystem to another photosystem, but these electrons can't come back to their original location. Means 680 photosystem 680 excited, and they passes their electron towards the electron acceptor. They passes their electron towards the electron transport system, and electron transport system passes their electron towards the P700, and P700 passes their electron towards the 700 electron acceptor, and they make energy. So electron flow is this kind of zigzag manner. But in the case of cyclic photophosphorylation, look at here. Now what kind of phenomena? In cyclic photophosphorylation, only photosystem 1 can participate. Because of cyclic photophosphorylation, we need only ATP. Here we get ATP, guys. Here we get ATP, and here we get NADPH. Now after accepting electron, they release ATP molecule, and yes, this molecule we want. Plant want this molecule. And after excitation with electron transport system, they pass their electron towards the P700 photosystem 1, and they activate, and again they pass their electron towards the electron transport system. So this mechanism is occur. So electron rotation like this. So this is the cyclic photophosphorylation. No breakdown of H2O molecule, no breakdown of photosystem second, no requirement of other thing. They reproduce ATP, 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 ATP. They generate ATP, ATP, ATP. When plant wants ATP, then they produce cyclic. And when plant want NADPH, what they do if they want to do H2O breakdown, so they do Z scheme, non-cyclic photophosphorylation. Now let's discuss about cyclic photophosphorylation in detail. Living organisms have a capability of extracting energy from the oxidable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. Means ATP, overall they want to say that ATP, this is the molecule that carry energy. The process through which ATP is synthesized by cell in mitochondria and chloroplast, that's it, is named phosphorylation. When ATP is synthesized, yes, in non-cyclic also ATP is synthesized, guys, but in a less amount because electron can't come back. Photophosphorylation is the synthesis of ATP from ATP, means ATP from ATP, and inorganic phosphate. This is the inorganic phosphate in the presence of light. Yes, we already discussed this thing. When the two phosphosystem work in a series, first photosystem second and then photosystem first, a process called non-cyclic photophosphorylation we discussed in our previous lecture. The two photosystems are connected through an electron transport chain. Yes, there is a electron transport system, a chain. Now, as seen earlier in the Z scheme, both ATP and NADPH are synthesized by this kind of electron flow. So, they are talking about non-cyclic photophosphorylation. When only PS1 means when only photosystem 1 is functional, the electron is circulate within the photosystem. Means this electron circulate within the photosystem, here to here, here to here, here to here, again. So this is the cyclic pattern, guys. This is the cyclic pattern, and the phosphorylation occurred due to the cyclic flow of electron. Phosphorylation means ATP's formation. So now ATP formation occur by this cyclic flow. ATP. So a possible location where this could be happening in the stroma lamellae. Yes, this phenomena occur, but where is stroma lamellae? We know now this is the thylakoid structure, and this is the extended thylakoid, and this is the different set of. Look at here, guys. So photosystem, of course, photosystem 1 also present here. Photosystem 2 present here. So this cyclic phenomena occur here. They produce ATP and ATP. While the member or lamellae of the grana have both photosystem 1 and photosystem 2, it does not mean that photosystem 2 not here. Yes, this is also here, guys. Stroma lamellae member lack photosystem 2 as well as NADPH reductase. They want to say that yes, photosystem 2 is here but less in number, or sometimes negligible, but here they are lots of in number, but here photosystem 1 is not present. So the excitation electron does not pass on the NADPH, but it cyclic back to photosystem 1 complex. They want to say that according to non-cyclic scheme, here is the end product of NADPH. Here at the end product of cyclic photophosphorylation, end product is ATP formation. This step is absent, step is absent, and this step is also absent. They want to say that. Here, look at here, guys. Look at here. Light absorbed by P700. What is the wavelength of light? They absorbed 700 nm. And here also electron is coming here, and they excited, and they passes their electron towards the electron acceptor, and electron acceptor get their electron and activate electron transport system, and ATP will produce. And again electron transport system activate, they passes their electron towards the photosystem first, and this is the whole mechanism. This is the whole mechanism. This is the whole mechanism. So electron move in a cyclic way. Cyclic way. Due to cyclic photophosphorylation, we get lots of ATP, and due to non-cyclic photophosphorylation, we also get ATP, but compared than this, we get less ATP, and we also get NADPH. But in cyclic, we can't get any kind of NADPH. No. In this system, in this cyclic system, photosystem 2 does not take participation. No. Here, photosystem 1 and photosystem 2 both are take participation. High ATP form. Yes, high ATP form. Here less ATP is formed. Yes. So this is the major difference between non-cyclic and cyclic photophosphorylation. Let's switch towards the chemo-osmotic hypothesis. In chemo-osmotic hypothesis, this hypothesis again explained non-cyclic and cyclic photophosphorylation. They said again a new term, proton gradient. H plus. They increase in lumen of thylakoid membrane. Yes, guys, again they understand, again they describe about non-cyclic and cyclic photophosphorylation and some protein gradient stuff. Let's start, guys. Let's start again. Since splitting of the water molecule take place on the inner side of the membrane, we know now, guys, one water molecule will break and they form 2 hydrogen ion plus 2 electron and half molecule of oxygen. Now, the proton or hydrogen ions that are produced by the splitting of the water accumulate within the lumen of the thylakoid. Yes, this phenomena where is occur? Here is the one thylakoid. Here is photosystem second is present. Here the photosystem first is present. And here breakdown of water take participate. Here the breakdown of water participate. Then they produce some hydrogen ion and then some protons and half molecule of oxygen where lumen of the thylakoid membrane. They only want what they want. They only want to increase positive charge in thylakoid lumen. Look at here. I explain here. Wow, good. This is the photosystem. This is the photosystem second. This is the photosystem second. This is the photosystem second, and this is the photosystem 1. I'm repeating again. Here is photosystem second P680, and here is photosystem 1 P700. This term is clear. They get light. Which wavelength? 680 nms wavelength. After exposure with this much energy, they excited. Their electron is excited. Now this term is clear. Their electron is excited. This is the electron. And here in thylakoid lumen, oxidation of water means one water molecule will break, and half what oxygen will form, H plus will form, and electron passes towards the photosystem. Clear, guys? I know this is clear. Now electron moves towards the out. They accepted by, I told you na, some electron transport system. Here they describe the elaborate manner. First, plastoquinone, they get electron. Plastoquinone, they take hydrogen from surrounding. And change into plastoquinone. They get hydrogen and change into this form. It does not require this thing, but they absorb. They take this H plus ion and they push towards the lumen side of thylakoid. Again, this hydrogen push the electron towards the thylakoid of membrane. Now electron is important. Then electron transfer towards the cytochrome B6 complex. This is the part of electron transport system. And this cytochrome B6, they pass their electron towards the plastocyanin. Here plastoquinone, they passes their electron towards the cytochrome B6F, and B6F passes their electron plastocyanin, and plastocyanin passes their electron towards the P700. Look at here. And P700 passes their electron towards the FD ferredoxin, and they passes their electron towards the FNR, and this energy is formed. So here again a plus ion is formed. So in outer medium, plus ion is increased, and they goes inside thylakoid of lumen. Here H plus is increased because of this electron transportation. So medium is, look at here na, lumen, high H plus, so acidic medium. And now they goes towards the where? They goes towards the F node F1 particle. And what they do? They enter in this ATP synthesis and they form ATP. This synthase produce what? ATP. So here H plus is high in concentration and they goes towards their low concentration. So this is the protein gradient. This is the potential gradient. This is the high electrochemical potential gradient. I repeat again by the flow chart, guys. Light come 680 nm and this will activate. They passes their electron towards the plastoquinone. Plastoquinone passes their electron towards the cytochrome B6F. Cytochrome B6F passes their electron towards the plastocyanin. Plastocyanin passes their electron towards the where guys? Where? P700, and they get activated. They passes their electron towards the ferredoxine. They passes their electron towards the F and R, and they finally produce what? They finally produce what? NADP plus 2 NADPH plus N. So this is the whole mechanism, guys. Also 700 nms light, they get absorbed. Okay, so this is the whole mechanism of chemosmosis. This is the mechanism of chemosmosis. And here what they want to say, that lumen of thylakoid contain high H plus ion. So lumen become acidic. Then H plus goes towards ATP synthesis, and ATP will produce by ATP synthase. So this is the ATP synthesis through chemo osmosis. And here last bullet is, as electrons move through the photosystem, protons are transported across the membrane. The whole mechanism, guys. This is the whole mechanism. Here NADP reductase enzyme is located on the stroma side of the membrane. Yes, guys. This conversion done by NADP reductase. This conversion to this conversion. This is not normal, na? This is the less energy form, and this is the highest energy form. This is done by NADP reductase. And where it located? Stroma part, guys. This is the thylakoid. This is the one thylakoid, na, guys. This is the one thylakoid. This is the set one thylakoid. This is the first thylakoid. Clear, guys? And another thylakoid is here, and another. But surrounding part is what? Stroma. So this mechanism is going to happen in stroma part. Only this. Come on, guys. All clear? All clear, guys? All clear? Now it's clear, guys. Now it's clear. If someone asked difference between non-cyclic and cycling, what you say? What you say? You have to clear multiple terms. In non-cyclic photophosphorylation, you have to remember their pathway. What? Light come. How much wavelength? 680 nms. And they embedded on 680 photosystem. Second, their electron excited, and they get by electron acceptor. Here now, guys, I'm repeating again, guys, be focused. Electron transfer towards the electron acceptor. Electron acceptor take their electron. Electron acceptor take their electron. How photosystem second is activated? How? Because they contain lots of photosynthetic pigments. They contain lots of photosynthetic pigments, and they absorb energy, and transport their electron here to here, here to here, and here to here, and these electron transfer towards the here. Clear, guys? Now electron acceptor collect all the electron, but they require more energy. So breakdown of one water molecule is here. So this is collect in the lumen part of the thylakoid, and they goes towards the transportation. Look at here. So multiple electron collect by electron acceptor, and then they pass. Now they pass their electron towards the wire, plastoquinone. Now after plastoquinone acceptation of electron, plastoquinone passes their electron towards the cytochrome B6F, and cytochrome B6F, they produce what? They produce ATP from ADP. They produce ATP, guys. Now okay. Now cytochrome B6 passes their electrons here. Phosphate, inorganic phosphate also form. So after excitation with cytochrome B6F, they passes their electron towards the plastocyanine. They have cyanine. Now plastocyanine passes their electron towards the P700. So they get their electron, and they also get some light. Wavelength is 700 nm, and they activate, and electron is transported by where? electron acceptor. And what they do now, they passes their electron towards the ferredoxine, and ferredoxine passes their electron towards the F and F and R produce energy molecule. Clear, guys? Now it's purely clear. And someone asked the what is the cyclic pathway? Then you have to say that you have to clear this flow chart, guys. This is not a shortcut. You describe all the thing, they get this much light and they break down of the water and electron transport here to here in non-cyclic. And if someone is asked what is the cyclic difference between cyclic, then what you say that in this system only photosystem one is participate, only this photosystem take participation, they absorb 700 nms light and they excited, they passes their electron towards the electron acceptor. Here is no breakdown of energy molecule. They passes their electron towards the cytochrome B6F and they passes the electron plastocyanin. And this is phenomena, this and this. These phenomena is happening in cyclic photophosphorylation. And here they produce continuously ATP from ADP. They continuously produce. So in cyclic pathway we get lots of ATP, but in non-cyclic pathway we get ATP and NADPH also. So that's all about our quick revision and chemo osmosis theory hypothesis. We complete this thing and this is the quick revision. Okay, now we produce ATP, means plant produce ATP. Yes, we saw they produce ATP and NADPH also. Yes, we already saw. Yes, NADPH is produced where the membrane of thylakoid. Here ATP produced and in stroma part NADPH is produced where they function, where they do their work. Yes, they do their work in dark reaction, and dark reaction happens in where they happens in stroma. Previously we discussed about light reaction and where they occur thylakoid's membrane by photosystem second by photosystem 1. We already discussed non-cyclic pathway and cyclic pathway, and here we were going to discuss about dark reaction. In dark reaction we study some cycles, how food is produced. Alright guys, in next video we will discuss about how ADP and NADPH used in biosynthetic phase, how they produce food after getting ATP and NADPH. In our next video we will start with Calvin cycle. Look at here, this is the Calvin cycle. We're going to discuss about Calvin cycle in our next video. So that's all for now. If you still not subscribe my channel, then subscribe guys. And thank you for listening me. Keep smiling and revise. Your exam is near. Revise.